Yeah, I think a few other people are here.
Okay, so can you see me?
No, I cannot.
So I have to, Christ, what do I have to do here?
There should be a button that looks like a video camera somewhere on your screen.
Yeah, so is it perhaps it's this one. No,
The one that's second from the left.
yeah, okay, I just hit that. Yeah, okay. Well, I've never done
There
this
it is.
before. Okay, good.
Yeah, there it is.
There it is, and I can see Clinton. Can't see anyone else, but that's
Yeah, I think everyone else is being not showing a video or something.
okay. So it's not very clear when Clinton talks. So if I'm not very clear, you'll stop me, I hope. All right?
Okay.
So, should I start?
Yeah.
Okay. So I had one email, and I had one. There's a lot of noise going on. Is it all right? I'm sorry. At this end, it sounds very noisy.
Yeah, sorry, I just muted everyone's mics.
Okay. Okay, that's fine. That's fine. So I had one email, and I just thought I better cover that. People were worried about what was happening when the T cell receptor for the alpha gene for the alpha chain is rearranged. When you do a V alpha to J alpha joining, you get rid of the intervening DNA. And actually, in the case of the T cell receptor, the delta genes are between V alpha and J alpha. And so you eliminate the possibility of making delta chains when you join V alpha to J alpha. And so normally you think of V alpha or as a V bank of genes and the J being close together, which they normally are, but in this case, they're far apart. And so you lose the ability to make delta chains when you make an alpha chain. So if that's all right, I'll go ahead. Last time we talked quite a bit about Teach1 and Teach2 cells. We had seen already in the past that when T cells respond to antigen, the antigen has to be processed and presented. And that came at a time when people had in their mind that B cells and T cells interacted through both the B cell and the T cell recognizing the antigen in the antigen bridge model, which we covered a long time back. So there the T cell receptor is recognizing nominal antigen and not a peptide bound to an MHC molecule. And so that model, the antigen bridge model of the B cell, T cell interacting, had to be changed. I'm just going to turn off the coffee, sorry. And so if we go to page 168, you can see what people came to. They abandoned the antigen bridge model because it's inconsistent with what people had learned about the T cell receptor. And in this case, the B cell, if we think of a hapten indicated by the arrow attached to a carrier, the hapten carrier conjugate bound to an anti-hapten B cell took that antigen into a compartment where it was processed. In other words, cut up into bits, and then newly synthesized class II molecules were directed to that compartment, picked up any peptides that might be able to bind to the class II molecule, and then the class II molecule was directed to the surface of the cell, and the T helper cell could now recognize the peptide class II MHC complex, and it generated and delivered a signal 2 to the B cell. So are there any questions or comments on that? It's very helpful to me if people say it's okay or something. Is that okay?
Sounds good to me.
Okay, good.
Yeah.
So, so then people wanted to understand how these cells interact. And, oh dear, this is where I make a mess of things. And they found out in various ways, and I'll just give you one indication, that if you go to figure 2 on page 169, the T helper cell, it turns out, has a molecule on its surface called CD40 ligand, and the B cell has CD40 on its surface, and the T helper cell, on the engagement of CD40 with CD40 ligand, sends signals to the B cell directly and via cytokines. And one of the pieces of evidence for this model is that there are human diseases in which people are deficient in CD40 ligand or in CD40, and they don't make antibody responses. And there was direct, and also if you add certain antibodies to CD40 on the B cell surface, so antibodies to CD40 on the B cell surface, you can mimic the signal given by the T helper cell. So are there any questions on that? So, so to summarize, we see that there's CD4 cells can help B cells, to activate B cells. People then looked at the requirements to activate CD8 cells. And in the next section, one system which is very important in development of concepts in the field, which was there was a class 1-like molecule which exists in allelic forms, and the molecule is called QA1. So I'm on page 169, and QA1b is an allele of QA1, and people found out that they could, in certain circumstances, raise cytotoxic T cells, which could attack QA1b bearing grafts. But what they found out is they took QA1b by itself, they could not actually prime for such cytotoxic T cells. So what they found out was that they could develop a hapten carrier system, just as we've seen a hapten carrier system for B cells, like in non-responder guinea pigs. PLL is the hapten, and you do not get a response to poly-A lysine in non-responders unless you couple the PLL to an immunogenic carrier. Case, what they found out is that a potential or something that would function as a carrier was a male antigen. So what they did is they took spleen cells from a QA1B mouse into another mouse where QA1B was not self, but they were otherwise genetically identical. And if they immunized with a female mouse with male spleen containing QA1B, they got priming. But if they took female spleen, which had just QA1B, and spleen cells, male spleen cells from a Q1A mouse, so that the two antigens, hapten and carrier, were on separate cells, they didn't get priming. So this made it look as though there was a great similarity in the activation of CD8 cells as there is for B cells, and it went further in ways which I won't is partially described in the notes, but I won't go into fully. They found out if they took QA1B, so they only the hapten, they would ablate the CD8 cells, which was specific for QA1B. So if they first of all gave female cells with QA1B, and then came on later with male cells with QA1B and HY, they could no longer prime any CD8 cells for QA1B because they weren't there anymore. So this suggested that in the absence of help, the hapten would inactivate the CD8 cell. And we'll see that's also true in the case of B cells. So that was a big package there. Does anyone want me to go through something again or clarify something about that?
Can you go through the last part again?
Yes.
Thank you.
In the first part, what we were seeing was that the QA1B hapten, you couldn't prime with that hapten unless you had HY, the male antigen, on the same cell. So the question arises, what happens if you give the QA1B by itself and you don't see anything happening because you don't get priming? But actually, you can find out that something happens because if you give QA1B first and then come back with QA1B HY, you no longer can prime because the CD8 cells have been wiped out. So not only do you require helper cells specific for HY, people showed actually that they were CD4 cells that had to recognize the HY antigen. If you don't have that help, not only do you not get activation, but the hapten by itself, the QA1B, will then activate the CD4 the CD8 T cells specific for QA1B. Did that make sense?
Yeah.
Okay. Good. So so now we're going to actually so to really put this into perspective, we've now seen that to activate CD8 cells, you need CD4 cells. In the absence of CD4 cells, antigen can inactivate CD8 cells. We've seen that to activate B cells, you require CD4 cells, and I'm telling you that we're going to shortly cover the fact that in the absence of CD4 cells, antigen will inactivate CD8 cells, B cells as well. So whether you have CD4 cells around determines the fate of other lymphocytes. CD4 cells around, B cells and T cells are inactivated. So CD4 cells are really critical, and whether you can activate them determines the consequences for the other lymphocytes. So CD4 activation And what controls whether antigen activates or inactivates CD4 cells is one of the most fundamental questions in immunology. Do people understand why I'm saying it's so important?
Yeah.
Yeah, okay. Okay, so we're going to go into models of activation of CD4 cells, but these models the end of the story. We're going to come back to this later. But what people actually found out was that we've seen that you need for antigen to be presented to a T cell. The antigen, if it's a protein antigen, needs to be processed and presented by an antigen presenting cell. And if you go to page 171, figure 3, people found out that the antigen presenting cell does not only have a peptide on MHC, class Q molecule, giving a signal, signal 1, but that actually the antigen presenting cell also has another molecule on its surface called a co-stimulatory molecule, and there's a receptor for the co-stimulatory molecule on the T cell, and that interaction is called the co-stimulatory signal, and you need that to get activation of the CD4 cell. So people realized that there was actually a two-signal model for the activation of the T cell. There's a two-signal model for the activation of a B cell, as we've seen, and you require help to activate a CD8 cell. So there's a two-signal model for the activation of a CD8 cell. But one thing I would point out, which will become important later, is that in order to activate a B cell, you need another antigen-specific cell, the T helper cell. To activate the CD8 cell, you need another antigen-specific cell, the CD4 cell. But in this model here, which honestly is a very good, important stepping stone, but was realized by people to be inadequate, the second signal does not require a second antigen-specific lymphocyte. It is a molecule on the antigen-presenting cell called the co-stimulatory molecule, and a lot of antigen-presenting cells have this molecule on. The prototypical type is called B7. It comes in different forms, B7.1 and B7.2, and the counter receptor on the T cell is CD28. So this is a model. I'll just give you a little indication of how it occurred, how it was realized. When people did transplants across MHC barriers, they found out that they got rejection, as we've seen, in definitely less than 20 days, usually less than 10. But they found out that if they did something to the transplant, culturing it in high oxygen tension, it turns out, for a few days, they then could transplant it across MHC barriers, and it was no longer rejected rapidly, only after months. And they realized that culturing it at high oxygen tension destroyed a passenger cell in the graft. And it turns out that these passenger cells, passenger leukocytes as they were called, are actually APC. And that was really proven, that whole transplant story, is if you take a graft, they were often thyroid grafts, cultured it at high oxygen tension, and then gave it to a recipient, which for which the thyroid graft was allogeneic, MCIM compatible. You didn't get rejection after the culturing it for a few days in high oxygen tension. But if you gave back from the donor APC, you got very smart rejection, showing that it was because you got rid of the APC in the graft by culturing it high oxygen tension. So this and various other observations gave rise to this model for how you activate CD4 cells. It was a 2-signal model, and all models that we will subsequently discuss are based on this model, on this 2-signal model. So it's a very important stage in the history of concepts. Are there any questions on that?
Not for me.
Okay, so that's helpful. Let's see, 170. So now there is something that we've avoided talking about, but perhaps it's not so, I mean, I'm sure a lot of you know about it, and that is that there are also cells which inhibit different kinds of immune responses. And on page 172, we've got an example of such a situation. This was a situation where if you give an antigen with an adjuvant, we've seen that adjuvants can be very important in getting responses. You get a very nice response. But if you give the antigen, in this case a carrier, KLH, without adjuvant, you don't get a response. But what's more is if you take a donor, immunize it with KLH without adjuvant, and then give it cells to a recipient, which you then challenge with DNP KLH in adjuvant, it inhibits the response to the hapten, the primary response to the hapten. And so these cells from the mouse given KLH without adjuvant were called suppressor cells or inhibitory cells. They are antigen specific, and they are T cells because you can get rid of them by treating the cells before you give them to the recipient with anti-Thy1 and complement. So these inhibitory T cells are going to be very important. Do people understand the assay in figure 4 for the inhibitory cells?
Can you just go over again why the cells that are why they inhibit the reaction with the hapten?
Yeah, okay. No reason. No reason that is known at this stage of the understanding. But let me put it another way, which might help. Suppose we give DNP KLH with adjuvant and we get a good response. And we give KLH without adjuvant. Let's say we give DNP KLH without adjuvant and we get no response. We're not sure whether that's because we didn't have something needed to get a response, or whether it was because giving KLH without adjuvant resulted in something which turned off a response. So not only did we not get a response, but we generated something which turned off a response. And to distinguish between those, people gave KLH without adjuvant and then found out that they had something there, which if they gave it to an animal which would otherwise make a response, prevented a response from happening to a hapten conjugated to the carrier. So how that works out, we will address down the road.
Okay.
But the point is, if you don't get a response, it could be because you have something which turns off a response, or it's because you lack something required to get a response, or could be both. So these suppressor cells became very important, and they are incredibly actually important. There's been a lot of controversy about them. Because there was there were hundreds of papers published, and by some very famous people, which were completely unreproducible. One of them got the Nobel Prize, and he probably published 50 papers on suppressor cells, and they all turn out to be wrong. It's sort of, I'm saying this because often when people are learning from textbooks, they think everything in the textbook is right. This is a terrible story. Obviously, the gentleman who got the Nobel Prize got it before he realized that all these things were wrong. He got it for something else, and finding out that these things were wrong. So that in the literature, people don't use the word suppressor cell anymore, but they call them regulatory cells or inhibitory cells. But in the classical literature, a lot of good classical literature shows that there are suppressor cells. It's just that some of the work on suppressor cells was actually wrong and very clearly shown to be wrong. So I'm not saying all the work on suppressor cells that I'm telling you about is wrong. Actually, I'm telling you that the things I'm telling you about suppressor cells are, I believe, really correct and standing the test of time. But now I'm going to try and do something which is you won't find in textbooks, but I think it's really, really important to make sense of things. And that is to try and take a bird's eye view and make a list for ourselves of all the conditions where we get suppressor cells for an antibody response. And if you look at pages 175, page 174 to 175, I list 7 conditions under which you get suppressor cells. One is when you give antigen around the time or shortly after birth, when people thought that animals were not yet immunologically competent to respond, or if you take an animal which is able to respond, you find out a medium dose will give you a good antibody response, but if you give lower doses, higher doses, you don't get an antibody response, and actually you can get suppressor cells generated. And there are a whole list, as I said, 7. I'm only going to make one point about this, which I'm going to come back to later. And that is that the first 5 conditions under which you get suppressor cells for antibody turn out to be the very same 5 conditions if you try to take a bird's eye view of the literature and you look at conditions under which you get cell-mediated immunity. So that suggests, we'll see when we try to integrate everything up together, that these 5 conditions are ones where you get cell-mediated immunity, and when you get cell-mediated immunity, you can get inhibitory cells which inhibit an antibody response. The 2 other conditions I'll just briefly deal with, but we will come back to them later again. One is that if you immunize with an antigen, or orally by putting it into the drinking water, for example, of mice, you'll find out that soon you will not be able to make any cell-mediated or antibody response to that antigen of most kinds of antibody. But actually, when you expose a mouse for an extended period of time to antigen through the oral route, you get IgA antibody produced. An IgA antibody is interesting from many points of view. One is that it doesn't fix complement. It doesn't bind to mast cells and cause acute inflammation. In fact, it's a very, very benign antibody. In fact, it's almost like it can counteract, block other antibodies from functioning if it's present in large amounts by binding to the antigen. So what people think is that you don't want to make inflammatory responses to your gut flora, which are often of great advantage to you. And so you make a kind of immunity which isn't antagonist to an invader, but it's more like a comfortable relationship where the IgA produced and downregulation of the other responses allows the gut flora to survive to their and your benefit. Are there any questions on that? Does it make sort of biological sense, what I'm saying?
Yeah.
Yeah, okay. So lastly, but not least, people found out that if they thymectomized certain strains of mice at 3 days of age, so they whipped out the thymus at 3 days of age, and they kept those mice for 2 to 3 months, they started coming down with autoimmunity. And that suggested that there were cells coming out of the thymus after 3 days in a normal mouse, which prevented autoimmunity. So what they did is they took a mouse, whipped out the thymus at 3 days. That means they would come down with autoimmunity. And then they looked for cells in the thymus, which were in the thymus of a mouse which was older than 3 days, let's say 10 days. And they found out that there were cells there which would suppress the autoimmunity. And these thymic cells had one marker on the surface, which was different from most other T cells. And that was it had a marker, a CD25 antigen, which is actually is a receptor, a high affinity receptor for IL-2. And they became known as natural T regulatory cells. So this gave rise to the idea that there are cells present in the thymus which have CD25 on their surface, which prevent autoimmunity. So that's quite a lot of information I've given you. And we will come back to these CD25 regulatory T cells. Yeah.
Go through the last 7 point once again quickly.
Yes. So to summarize, if you whip out the thymus at day 3 of life, you get autoimmunity. If you take such a mouse, but before waiting for 3 months and the autoimmunity arrives, at day 10, you give it thymocytes from mice which are older, 10 days old. You find out you can suppress the autoimmunity, and that suppression is due to thymocytes bearing the CD25 antigen. And we will come back to these regulatory T cells later on. It turns out that they usually produce IL-10 and TGF-beta. And that will become important, but we're not going to emphasize it now because it'll only become apparent later on why that's important. So the first, what I would say is the first 5 conditions under which you get suppressor cells for antibody are also conditions where you get a cell-mediated response. And I personally make sense of all this by saying that when you've got a very strong cell-mediated response, you can inhibit the antibody response. Then there are 2 other situations, that thymectomy situation, and then oral immunization where you get IgA produced at the expense of all other classes of immunity. And IgA is very benign in that it doesn't activate complement or acute inflammation like IgE, mast cell degranulation, and it allows bacterial flora to function and not be attacked, and us to make use of our bacterial flora. I'm sorry, I can't. I don't know whether I said the right thing, but I was trying to summarize again what I said. Are there any questions? All right. So the properties of the antigen-specific suppressor cells are, first of all, they've got the CD8 marker, the same marker as on cytotoxic T cells. So in the periphery, all T cells have either CD4 cells, the CD4 marker, the CD8 marker. There are no T cells in the periphery which have both. And there are a small number of T cells which have neither CD4 nor CD8, and those are the gamma delta cells we've discussed. The CD4 cells are class 2 restricted, the CD8 cells are class 1 restricted. So the CD4 cells, the CD8 cells are suppressors. So someone asked, how do these suppressor cells work? Well, as an introduction to that, if you remember how B cells and T cells work in aiding the antibody response, in order for T cells, which are specific for KLH, or as we now know, for peptide of KLH bound to a class 2 molecule, but T cells specific for KLH can only help a hapten-specific B cell if the hapten is attached to KLH. And we can understand that by the fact that if we have DNP KLH, the B cell, which is binding the DNP, look at figure 6b on page 177, the B cell will take in the DNP KLH, process the KLH peptides will bind to class 2, which will be expressed on the B cell surface, and then the T cell helper cell receptor can recognize the peptide class 2 complex and deliver, if it's activated, signal to CD40 ligand and cytokines. So I'm just saying that all that is going on in this figure. So what people did is they took an irradiated animal, gave it cells primed to DNP, cells primed to KLH, and then they would give DNP KLH, and they would get a good response to DNP, which is group 1 of this figure on page 176. Without a label of what number it is. So that's just roughly what we already know from past things. That's a way of getting a good response to a hapten. You have to have cells primed to the hapten and to the carrier. So now what they did is they had cells which had come from an animal which had been given human gamma globulin in a way that generated suppressor cells specific for human gamma globulin. And they knew that from the experiments we've already looked at the beginning of this session. But they wanted to work out something about the mechanism. So what they did is they found out that they could get suppression if they gave suppressor cells, C group 4, and they gave DNP HGG. So how they interpreted that in an antigen bridge type model, which we know isn't exactly right, but let's start off there because it gets pretty complicated. If you look in the figure 6b, you've got TSAB specific for HGG. The T cell receptor binds HGG, and the B cell binds the DNP, and you get a negative signal to the B cell. And other evidence, which we're just going to go into, is consistent with this model of how the suppressor cell gives a negative signal to the B cell. But do people just see how that, given group 4, how that's a possible explanation of group 4? So you've got suppressor cells specific for HGG. We're going to do a simple antigen bridge model. So we're not saying, we're pretending that the TSAB doesn't recognize a peptide in the context of an MHC molecule, but actually recognizes the antigen itself. And then the suppressor cell can inhibit the B cell from responding if there's link recognition. The B cell binding the DNP and the T suppressor cell binding HGG. Does that make some sense?
Yeah.
I mean, this is getting pretty hairy, actually. So if you don't have the DNP on the HGG in group 3, you don't get the negative signal, which show because the TSAB anti-HGG cell is not focused in its delivery of its negative signal to the B cell. And if you don't have the suppressor cell, group 2, you don't get inhibition of the response. So you require the suppressor cell, you require the DNP to be linked to HGG, which fits in with this model. So this is a way that people try to understand how suppressor cells act to inhibit an antibody response. It's obviously not consistent with everything we know because a CD8 cell recognizes a peptide bound to a class 1 molecule. And if you're really worried about that, the way of making it consistent is that the DNP HGG goes into the B cell, it's processed, and then expressed on the surface with a peptide derived from HGG binding to a class 1 molecule, and the T suppressor cell recognizes that and gives a suppressive signal. But that's beyond really what I think we should talk about. But if you are a person who really likes consistent pictures, which is a very good property to have, you want to know how different things can come together, and an antigen bridge model is really not viable with all that we know.
Would so in that example, would the suppressor signal still go to the B cell then, or to the other cell, the MHC2?
No, I mean, in this case, it would go to the B cell.
Yeah.
If there's no DNP on the HGG, it would work on a B cell which is specific for HGG.
Yeah.
All right? If the way that these suppressor cells work is way beyond the scope of what we should get into.
Okay.
All right?
Just rework.
Yep. Yep. So the last thing that I think we might talk about today is this is a way of negatively regulating B cell responses. I personally believe, and this is not generally accepted, but it's the only way I can make sense, tie things together, is actually these CD8 cells that we're talking about they're generated under 5 different conditions, which are listed under the conditions where you get cell-mediated immunity. So I think this is a mechanism where when you get a very strong cell-mediated response, you turn off the antibody response, just to put it into a context. So the last thing is, for today, is that people did experiments which led to the conclusion that if antigen interacts with a B cell without a T helper cell, that B cell was inactivated. And it had to interact with that B cell for about 2 days before it became irreversibly inactivated. So that's a conclusion, and the first piece of evidence for it was from something called the spleen focus assay, which is on page 177. And I'm going to just give you the scenario, I think, of how the person who did this experiment, what he had in mind. So what he did is, it turns out that when you activate helper cells, once you've actually activated them, you can irradiate them. They will still help. And that's not really very surprising, because if you think about clonal selection theory, people knew that that making a primary response by an animal was very sensitive to radiation, and they came to the conclusion that very scarce cells, when triggered, multiply up to make more factories that can make antibody. And once the B cells have multiplied up, once they're making antibody, you can irradiate antibody-producing cells, and they go on making antibody. So really, the sensitivity to radiation is because it damages a single molecule of DNA, and if you don't have time to repair and you're forced into cell division, you die. And so the sensitivity of making immune responses to radiation is because lymphocytes normally have to divide before they can give rise to an immune response. So what Kleinman did is he took mice and he immunized them with KLH so that they had helper cells in them, and then he irradiated them so that he ablated all the ability of any lymphocyte, apart from those induced helper cells, to function. And then he put in one day after irradiation, he put in a few B cells. He knew that B cells, bone marrow contained precursors of antibody-producing cells. And he put in rather few, a few thousand or 10,000. And then after 2 days, or 1 day, he took the spleen cell, the spleen intact, cut it up into 50 pieces, and put each piece into a well with DNP KLH. And what he found out is that most of the fragments, nothing happened. But occasionally, he found out that the fragments started making anti-DNP antibody in response to DNP KLH. And as the mice were primed to KLH, he thought that the bone marrow that he had given in had some anti-DNP precursor cells, and if one was lodged into that fragment, when he cut up the spleen into 50 parts, it would make antibody. And he actually showed that that was the case. He got so much antibody that he could do equilibrium dialysis, and it was very uniform. The antibody either had kappa or lambda chains. It was had uniform binding constant. He could get so much antibody. So he had actually a clone of B cells producing antibody in those fragments. And he could show that statistically, if he gave half the number of cells, he got half the number of fragments being positive. And he could show that he needed just one B cell to get a positive clone of B cells produced. So do people understand what's roughly happening in this assay?
I think so, yeah.
Okay, thank you. So what he then did is he took these fragments and he delayed giving the DNP KLH for 2 days. And he found out he still got positive responses. Delaying it for 2 days didn't matter. And then what he did is he gave DNP of albumin for those 2 days, and he couldn't get any antibody produced anymore. So therefore, he thought, there are helper cells specific for KLH, because I primed my mice with KLH. There are no helper cells specific for ovalbumin. So if the B cell that's there interacts with DNP ovalbumin for 2 days, it's wiped out. If he did it for only 1 day, they weren't wiped out so efficiently. So he concluded that B cells interacting with antigen without helper cells resulted in that B cell becoming non-functional. Did that make sense? Glinton, did you understand what I was saying?
Yep. I think
Okay.
I followed along fine. Too bad.
Okay, so we've got to the end of the time and people might have other classes and things. So I'll take it up from there on Monday, if that's all right. Okay, and I know I'm going through this fairly fast, but I'm trying to do it so that we can see a big picture in the end, because that's really where it gets exciting, to be quite honest. Okay.
Okay, thank you.
Thanks to everyone for participating.
Have a good day.
Thanks, Peter.
Thanks.
Bye.
Bye-bye.
Bye.